Hello everyone, today we are going to study signaling pathways. This is a part of signal transduction. As we know, plant cells constantly communicate with one another. Cell controls and coordinate their activity in response to signals. As we have studied earlier, in plant receptors, most of the protein act as a plant receptors which detects the stimuli and respond to it. So, signaling pathways are nothing but the series of or the network of reaction in response to receptions of the signal. Today in this module we are going to study various signaling pathways in plant system. The learning objective of these modules are introduction of signaling pathway, two component signaling system, Auxin signaling, calcium ion based signaling, G protein mediated signaling, stress signal transduction, cell division and differentiation signaling cascades. Introduction. In plants, similar to that in animals, cells are in constant communication with one another. Plant cells communicate to coordinate their activities in response to the changing condition of light, dark and temperature that guide the plant's cycle of growth, flowering and fruiting. Plant cells also communicate for coordination of their roots, stems and leaves activities. Elaborate signaling mechanisms are required in all complex multicellular organisms for both internal as well as external responses. Animals have two specialized systems for the purpose, for example, nervous and endocrine system, but in plants which lack motility, only hormonal system has developed which work as a chemical messengers. Plant signaling pathways differ in significant ways from those of animals in selection of transmembrane receptors, secondary messengers and dedicated mechanism to environmental cues such as stress, drought and light. Calcium, inositol phospholipids, G proteins, cyclic nucleotides, protein kinases and protein phosphatases are common molecules present in majority of plants' signaling pathways. G protein coupled receptors, though well characterized in animals and yeast plant, do not have evidence of comparative abundance of the same. Plants have signaling pathways involving the hydrolysis of phosphatidyl inositol 4,5-bisphosphate by plasma membrane associated phospholipase. Many signal transduction pathways came into being when plants are challenged with various environmental stresses. It is still very much inclusive as how to categorize these events. Specific networks for cold, drought and salt stresses can be divided into three major categories: osmotic or oxidative stress signaling that makes use of MAPK modules. Second, calcium dependent signaling that lead to the activation of LEA type genes such as DRE or CRT class of genes. And third, calcium dependent SOS signaling that regulates ion homeostasis. Stress signaling knowledge is vital for development of rational breeding and transgenic strategies to improve stress tolerance varieties. Cell differentiation in both shoot and root requires strict control of various cell populations, which is regulated by specific signaling mechanisms. Coordinating expression of genes during plant development as well as during response to environmental cues is required. Elaborate signaling mechanisms such internal as external signaling agent bring about their effects by means of sequences of biochemical reaction called signal transduction pathways. These pathways, due to cascading effect, greatly amplify the original signal and ultimately results in the activation or repression of genes. Signaling cascades involves generation of second messengers inside the cell that greatly amplify the original signals. Among the most common second messengers in plant signaling pathways are 3,5' cyclic AMP, 3,5 cyclic GMP, that is cGMP, nitric oxide, cyclic ADP ribose, 1,2-diacyl glycerol, that is DAG, inositol, 1,4,5 triphosphate, that is IP3, and calcium ion. Entire genomes of plants such as Arabidopsis and rice are now sequenced that leads to the possibility to search for unknown proteins by database mining. Such database searches of a plant sequence have successfully identified family of receptors like protein kinases, that is RLK. 3 types of RLKs have been identified in plants on the basis of extracellular domains. Primarily, plant development was known to be regulated by only 5 types of hormones: auxins, gibberellins, cytokinin, ethylene, and abscisic acid. However, with new set of evidences confirm presence of plant steroid hormones, the brassionosteroid, a variety of other signaling molecules having role in resistance to pathogens and defense against herbivores, are also been proved by experimentation, including jasponic acid, salicylic acid, and the polypeptide cysteamine. Phototropism is the phenomenon exclusive to plants resulted by differential growth due to bending towards light. Phototropism occurs by directional cell elongation, which is stimulated by the growth regulator auxin. Thus, plant signal transduction is diverse as animal system, yet to lack clear understanding of majority of the pathways. Signaling pathways. Plants are unique as compared to animals in the fact that their development and morphogenesis are plastic throughout their lives. They continuously monitor both biotic and abiotic parameters of their environment, and these sensory perceptions shapes their organs and bodies. Plants get their environmental cues via sensory perceptions, which are critical for their survival, especially light and gravity. 2 physical factors pervading the universe are essential in their respects. Despite having a relatively simple body organization, plants need sophisticated sets of coordinative processes of diverse functions such as root shoot coordination, coordination amongst the radical tissues, patterning in the totipotent plant cells, to name few. Auxin as a central in this respect, that regulates patterning of a cell differentiation, cell division and cell expansion. Plant cell to cell communication via various signaling cascades. In that, first, two component signaling system, second, Auxin signaling, third, calcium based signaling, G protein mediated signaling, stress signal transduction, cell division and differentiation signaling cascade, and last, abscisic acid based stomatal signaling. Next slide. Hormonal signaling. Various growth regulators, also called plant hormones, helps to coordinate plant developments. They include ethylene, auxin, cytokinin, gibberellin and abscisic acid. Growth regulators are small molecules made by most of plant cells. They diffuse readily through cell walls and can act locally or be transported to influence cell at a distance. Each growth regulator can have multiple effectors. The specific effect depends on various factors such as which other growth regulators are acting, or on environmental conditions, or on the nutritional state of the plant, or on the responsiveness of the target cells. Ethylene is an important plant hormone. This small gas molecule influences plant development in various ways, such as promotion of fruit ripening, leaf abscission and plant senescence. It also functions as a stress signal in response to wounding, infection and flooding. When the shoot of a germinating seedlings, for example, encounters an obstacle, it can respond in multiple ways, which is controlled by ethylene. Two component signaling system operates in bacteria, fungi as well as in plants, but apparently not in animals. When animals should have given up this way of signaling remains a mystery. In this figure shows a current view of ethylene two component signaling pathways in absence of ethylene. The receptors and the MAP kinase module are active, leading to inhibition of the gene regulatory proteins in the nucleus that are responsible for the transcription. Plant responses to endogenous signals as well as environmental cues are mediated by several hormones which are implicated in almost every aspect of plant growth. From seed germination to flowering, ubiquitin mediated proteolysis has been shown to regulate different steps of plant hormone signal transduction. The plant hormone auxins or indole 3 acetic acid is implicated in many developmental processes in plants. These hormones is also called a morphogen, as its local accumulation and directional flux in different plant tissues and organs. Ubiquitin mediated proteolysis has been shown to regulate different steps of plant hormones signal transduction. The plant hormone auxin or indole 3 acetic acid is implicated in many developmental processes in plants. This hormone is also called a morphogen, as its local accumulation and directional flux in the different plant tissues and organs determine cell fate and identity. It may also establish polarized growth and turn on many developmental pathways. The response to auxin is extremely rapid, involving cell growth and activation of a wide range of auxin responsive genes. In this figure, a current model of ubiquitin mediated proteolysis in auxin signal transduction is seen. In this, the absence of auxin and indole acetic acid protein, which is shown by blue balls, heteromerize with auxin response factor, that is ARF factor transcriptional activators, which is shown by purple balls, thereby preventing the latter from activating auxin inducible genes with linked upstream OX-ray cassettes auxin through an unknown mechanism, that is OX indole acetic acid protein phosphorylation, perhaps increases the binding of OXIA to the TIR1 subunit of the SCF TIR1 complex, which catalyzes the ubiquitination of auxin indole acetic acid proteins, targeting them for proteasome dependent degradation. OX indole acetic acid protein degradation releases ARF factor, which are now able to activate transcription of auxin inducible genes, including OX/indole acetic acid genes. The de novo or synthesis of auxin indole acetic acid proteins leads to a sequestration of ARF factors into inactive heterodimer. It is likely that RUB1 cycling might be important for SCFTRI1 complex functions in mediating auxin signal transduction. In addition to cell expansion, auxin can induce cell division, a characteristic widely employed for tissue culture and the generation of transgenic plants. Investigation of lateral root formation in Arabidopsis suggested that auxin regulates cell division. Lateral root formation in Arabidopsis suggested that auxin regulates cell division through a G protein dependent pathways. Bifurcation of auxin signaling appears to imply a differential interaction with the cytoskeleton. When the contributions of cell division and cell elongation were assessed separately, the natural auxin indole acetic acid along with NAA and the auxin transport inhibitor 235 triiodobenzoic acid, that is TIBA, were observed to inhibit cell elongation while leaving filamentous actin unaltered. In contrast, 2,4-D and the polar transport inhibitor NPA inhibited cell division and at the same time eliminated actin filaments. Figure shows model for bifurcation of auxin signaling in the regulation of cell division and cell elongation in tobacco cells according to Campanoni and Nick in 2005, modified according to Raman and al. 2007. The auxin receptor with the high affinity for 1-naphthalene acetic acid regulates cell elongation and independent of G protein activity and does not cause a disassembly of actin, whereas the auxin receptor with high affinity for 2,4-dichlorophenoxyacetic acid triggers a signal chain that involves the activity of G protein triggers disassembly of actin filaments. This signal chain is inhibited by pertussin toxin and activated by aluminum tetrafluoride. Both pathways are mutually inhibitory. Since the natural auxin, that is indole acetic acid, can enter the cell from any direction, but will exit in a defined direction which is governed by the localized activity of the efflux carrier, it can collect the input from several neighbors and focus this input into a clear directional output. It is this property that makes auxin a versatile and robust investigator for cell to cell communication in various plant signaling cascades. Calcium based signaling. The maintenance of intracellular ionic homeostasis is fundamental to the physiological well-being of a living cell. The calcium is a versatile signal ion regulating many physiological processes such as ionic balance, gene expression, metabolism of lipids, proteins and carbohydrates, cell growth, cell division, cell development and secretion. In plants, calcium ion plays many essential roles. It is an essential plant nutrient required for growth and development of the meristematic root and the shoot tip. Calcium ion helps in the formation of microtubules and microtubules which are essential for the anaphasic movement of chromosomes. Calcium ion is an important divalent cation and is a structural component of a cell wall and membranes, where it exists as calcium pectate. It is also required as a counter cation for inorganic and organic ions in the vacuoles and as an intracellular messenger in the cytosol. Several genes are reported to be upregulated in response to calcium in plants. An analysis of transcriptome changes revealed that 230 calcium responsive genes are present. During evolution, positively charged calcium was selected as the primary signaling element of cells. Calcium ion is one of the principal second messenger for functioning as a central node in the overall signaling web and plays an important role in providing stress tolerance to plants. Environmental or developmental stress leads to increased cytosolic calcium ion which initiates the stress signal transduction pathways for the stress tolerance. Calcium ion is the most tightly regulated ion within all membrane bound organisms across the phyla. Calcium ion binds to several proteins to affect changes in localization, association and function. Upregulation of calcium ion concentration in cytoplasm is found to be a key event in the plant cell for transduction of various signal to biological effect. This suggests that calcium ion is communication point in the cytoplasm for the crosstalk between different signal transduction pathways and finally helps in interchanging of the information. The calcium ion signaling involves role for many sensor proteins that decodes temporal and spatial changes in cellular calcium ion concentration. Figure shows calcium and SOS pathway in regulation of ion homeostasis by various ion pumps. Under saline condition, the increase in the cytosolic sodium level causes damage to several cellular processes. One of the major salt detoxifying mechanism in the cell is the calcium activated SOS3 or SOS2 protein complex, which activates SOS1 sodium ion hydrogen ion antiporter on the plasma membrane, responsible for extrusion of sodium ion from the cell. At the same time, SOS3 and SOS2 complexes is also involved in inhibiting HKT1, a low affinity potassium transporter which transports sodium ion under high salt condition. Recently, another member of SOS3 family, CBL10, has also been found to form a complex within SOS2. This complex is speculated to regulate both extrusion of sodium ion by regulating SOS1 and sequestration or compartmentalization of sodium ion into the vacuole. SOS1 also interacts with RCD1 and imparts protection against reactive oxygen species and oxidative stress. SOS2 also works to activate H plus ATPase under salt stress and helps in restraining ion homeostasis. G protein mediated signaling. Majority of the signals are mostly perceived at the level of membrane, and therefore transmembrane events are the likely routes for signal generation and transduction. In plants, the best characterized plasma membrane based receptors are two kinds: transmembrane receptor enzymes, usually kinase, or G protein coupled receptors, that is GPCRS. There are two kinds of G protein, monomeric and heterotrimeric G proteins are involved in the transmission of external signals via receptor molecules to effector molecules. The involvement of G proteins in various signals transduction pathways was initially implied pharmacologically using various agonist and antagonist involvement of G proteins in light signaling pathways has also been shown. Although the human genome contains about 1000 GPCRS, but only single gene, that is GCR1 encoding a putative GPCR has been identified in Arabidopsis thaliana, which has reported to be regulator of cell cycle and also involved in ABA signaling in guard cells. Using a different bioinformatics tool, it was shown that there might be as many as 390 divergent GPCR candidates in Arabidopsis. Based on sequence homology and functional similarity, the animal GPCRS can be grouped into several families such as family A, that is rhodopsin like, family B secretin like, family C metabotropic or pheromone like, family D fungal pheromone, family E camp receptors, and family F frizzles or smoothened family. Recently, though computational studies of family A and family B of GPCRS Vora has done by Vora and et al. In 2017 reported that the plant GCR1, that is GPCR, contained homology with family A and family B and family E of GPCRS. Receptor mediated signal transduction in animal system is either carried forward via the mechanism of protein-protein interaction or via generation of second messengers. However, this mode of signal operation has not been reported and well studied in plants. In plants, in majority of cases, the alternate mode of action, for example, to activate ionic channels in the membrane and two component signaling as well as the movement of receptors directly in the nucleus seems to be operative. The mechanism by which G proteins pass on the messages to downstream elements is not very clear in plant system. In a review by Auxin, it was mentioned that phospholipases might be regulated by G proteins. This figure shows schematic representation of possible role of heterotrimeric G proteins in providing salinity tolerance to plants. On basis of interaction of GA and PLC, the role of GA in salinity strength tolerance and the possible mechanism of salinity tolerance may be operated through PLC, whereas mechanism of heat tolerance still remain to discovered. Stress signal transduction. Plants are constantly exposed with many biotic and abiotic environmental stresses, which causes considerable losses of crop yield worldwide, whereas demand for feeding increasing population is always on rise. Recently, Vallée described that mechanical wounding stress also induce both the biotic and abiotic stress response via a novel cis element. In general, the plant does not follow exact same signaling cascades as in non-plant system, but contains several unique component and unique unexpected crosstalk among signaling components. With the complete sequence of Arabidopsis genome and its analysis, it seems that plants encodes a very large number of protein that may be involved in signal processing. This may be because of the fact that plants are sessile and therefore they have to very effectively pursue the environmental variables for their growth, development and adaptation. Also, the signaling has to be modified for a defensive mode of survival and not for offensive one. To meet these demands, they have on one hand retained some of the components of bacterial signal pathways, as for example, the two component receptors for hormones, and on the other hand have some components of higher eukaryotic animal systems. Knowledge about stress signal transduction is vital for continued development of strategies for rational breeding and transgenic varieties to improve stress tolerance in crops. Signal transduction relies on the proper spatial and temporal coordination of all signaling molecules for successful transmission of information. Drought, salt and cold stresses induce the accumulation of ROS such as superoxide, hydrogen peroxide and hydroxyl radical. During biotic and abiotic stress, plants produce increased amount of hormones such as ABA and ethylene. In addition, SA, perhaps jasmonic acid, may be involved in some parts of stress responses. These hormones may interact with one another in regulating stress signaling and plant stress tolerance. ABA is undoubtedly the plant hormone most intimately involved in stress signal transduction. Figure shows a generic pathway for the transduction of cold, drought and salt stresses signals in plants. Example of signaling components in each of the steps are shown. Secondary signaling molecule can cause receptor mediated calcium ion release, which indicated by the feedback arrow. Example of signaling partners that module the main pathway are also shown. These partners can be regulated by the main pathways. Signaling can also bypass calcium ion or secondary signaling molecule in early signaling steps. Genetic approaches are important tool for analyzing complex processes such as signal transduction during stress condition. Conventional genetic screens based on stress injury or tolerance phenotypes have been documented. However, due to functional redundancy of the pathways and its components in the control of plant stress tolerance, adoption of forward and reverse genetic approaches may certainly expedite the understanding of many evading concepts of plant signaling mechanism. This figure shows major type of signaling for plants during cold, drought and stresses. Representative cascades outputs biological functions and example of mutants with the phenotypes. Inactive of defects in the representative biological functions are shown. Type 1 signaling involves the generation of ROS, scavenging enzymes and antioxidant compounds as well as osmolytes. The involvement of MAPK pathways in the production of osmocytes in plants has not been demonstrated experimentally. Under osmotic stress, altered MAPK signaling may contribute to changed cell cycle regulation and growth retardation. Type 2 signaling involves the production of stress responsive protein, mostly undefined function. Arrows indicate the direction of signal flux. Primary sensors are shown to be localized in the membrane. Cell division and differentiation signaling cascades. To build an entire plant from a single cell require strict temporal as well as spatial control of stem cell populations as well as specification of appropriate cell fates. Once cell enter a differentiation program, it is now experimental proven fact that both intracellular and intercellular signaling play important roles in controlling cell division patterns and cell specification. The shoot apical meristem, that is SAM, is the source of cells for all aerial organs produced after germination. The SAM is first formed at the globular stage of embryogenesis and develops into a dome shapes in the mature embryo. In Arabidopsis, a stem cell population persists even after the transition from vegetative to reproductive growth, allowing the inflorescence SAM to produce an intermediate number of flowers on its flanks. The maintenance of stem cells requires WUS homodomain transcription factor, loss of function WUS Arabidopsis mutants have defects in this SAM at all developmental stages. Should meristemless, that is STM, Arabidopsis mutant exhibit defects in SAM formation, maintenance and similar to those of WUS. Mutation in their CLA VATA clavata that loci, that is CLV1, CLV2 and CLV3 results in phenotype opposite to those of WUS and STM stem. The CLV1 protein is a receptor like kinases composed of leucine rich repeat containing extracellular domain with a repetitive receptor function and cytoplasmic cell kinase domain linked through a transmembrane domain. Figure shows models of stem cell regulation. Stem cell maintenance in the vegetative and inflorescence meristem. The size of the stem cell population is controlled by a regulatory loop between WUS and CLV. When the number of stem cells is increased, more CLV3 ligand is released from the stem cells, which is perceived by CLV1 and CLV2 receptor kinases in underlying layers. This result in fewer cell expressing WUS, thereby attenuating stem cells promoting activity. In contrast, when the number of stem cells is decreased, less CLV ligand is released. Consequently, more cells start to express WUS, thereby promoting stem cells identity, which is shown in right. Termination of the stem cell population in early flower primordia stage 2, which is shown in left. The meristem identity gene LFY is expressed throughout the primordial, whereas WUS is expressed in the center to reserve a stem cell population for later developmental stages. Later in stage 3, LFY and WUS together activate AG expression in center of the primordia. As flower development proceeds, WUS expression becomes downregulated by AG. AG expression persists in the center of primordia and determines floral organ identities in whorls 3 and 4. Compared with the SAM, the root meristem RM has a fewer cell and a simpler structure. In Arabidopsis, the cell division pattern in the RM has been well characterized. The mature part of the Arabidopsis root is composed of concentrically organized cell layers, which from inside to outside form vasculature, pericycle, endodermis, cortex and epidermis. A laser ablation study first gave conclusive evidence for the importance of positional signaling in root pattern formation. The root initial cells can be thought of as functionally equivalent to CLV3 expressing system cells of the SAM. Next, abscisic acid based stomatal signaling. Abscisic acid ABA is a regulator of stomatal aperture via its effects on osmotically driven changes in guard cells turgor. Abscisic acid inhibits the H1 ATPase and activates both non-selective calcium ion permeable channel and a slow, that is S type ion channel. ABA signal transduction in the guard cell is hypothecated to mediated by changes in cytosolic free calcium ion concentration as well as calcium ion independent pathways. There is experimental evidence that transduction of ABA signal in guard cells is mediated by phospholipase. D. PLD activation. The mechanism of stomatal opening and closing are thought to operate through distinct signaling response elements. The role of PLD in ABA response is observed in guard cells of Vicia faba tobato, guard cells, protoplast, and also an ABA related senescence in Arabidopsis was documented. The ABA responses of these systems involves alteration in cellular processes ranging from gene expression to ion transporter activity. This involvement of PLD in ABA responses of disparate cell types and varied physiological processes suggests that these enzymes may be conserved element in the transduction of ABA signal in many types of plant cells. Next, summary. All organisms face a common challenges how to adapt successfully to changing environmental conditions. In the case of non-sessile plants, adapting to shading or reduced water availability usually involves making coordinated alterations to growth and development so as to ensure the optimum capture and use of available resources. There is need to integrate both genetic and biochemical approaches to study plant signal transduction. Although the use of mutants has been a powerful technique for identifying genes encoding components of signal transduction pathways, the way forward will clearly be to identify interacting proteins, as by analogy with animal system it is likely that individual components will be organized into supramolecular complexes. Among all the signaling molecules, no mutants lacking either auxins or cytokinin have been found, suggesting that mutants for either of these molecules are embryonically lethal. Auxin signaling act via MAP kinase mediated phosphorylation cascades of proteins leading to activation of transcription factors. Gibberellin signal transduction seems to involve G proteins and cyclic GMP, which leads to induction of amylase gene transcription via production of transcription factor GAMYB. Cytokinin signaling is initiated by binding to CRE1 receptor, thereby phosphorylating type A and B AAR proteins to mediate various cellular functions. Signal transduction of ethylene is via transmembrane receptor, which is encoded by a family of genes that encodes proteins similar to bacterial two-component system. Abscisic acid signaling involves coordinated action of positive as well as negative regulators, which affects many diverse processes. These include transcription, RNA processing, protein phosphorylation, protein farnesylation, and metabolism of secondary messengers. Abscisic acid response in any given cell type is found to be regulated by more than one signal transduction pathway, which provides necessary flexibility in response. The technical advances being made in proteomics, genomics coupled with mutant screens and classical biochemical approaches are going to help in understanding how plant perceives and responds to diverse signals. Thank you.